everyone, this is Amanda Moore from the Angelman Syndrome Foundation, and you're listening to the ASF Podcast, the show that explains everything you want to know about Angelman Syndrome. We're so excited you're here, and we hope you enjoy the show.
For this week's podcast, we're sharing a session from the 2024 ASF Family Conference. We hope you find this information helpful and enjoy the listen.
The way this will work best is if you stop me the minute you're like, what did you just say? I don't get it. Because trust me, if you don't get it, there are lots of other people who don't get it. And you can hear this 1,000 times and hear something new for the first time. I think we can all tell you stories about like when the doctor told us we were diagnosed, and I bet none of us got half of what the doctor said, right? Because you hear, my child just got diagnosed, and your brain kind of stops, and they keep talking, and you're like, whatever, dude. Or dudette, whoever it is. Okay, so my name is Rebecca Burdine. I am a professor of molecular biology at Princeton University. It sounds really highfalutin. It's not. This is my daughter, Sophie. Sophie is 18, thank you. She loves to ride her bike. If you put her in a gait trainer, she will do 3 miles at school in the gait trainer, especially if there are people to run into, very motivational. She has never taken an independent step in her life. She has never stood by herself. But in a bike, she's hell on wheels. Oh, sorry, there's young kids here. Sorry about that. I am on the ASF Board of Directors at the moment. I am the chair of the ASF Scientific Advisory Committee, and I am currently a board member for something called the Society for Developmental Biology. People, when they talk, should tell you these things because it tells you where their biases might be and whether or not they're telling you something that maybe is a little slanted. So everyone should do this. We're going to talk about the genetics of Angelman syndrome. This is really where I want you to put your hands up and make sure you understand it. We'll do a brief discussion about genotypes and phenotypes. Genotype is what your DNA looks like. Phenotype is how you present to the world. We'll talk about where to focus if you are just new to this journey, if you're just getting started, or maybe remind you where we should be focusing. So here's the first part that's important. You receive one set of chromosomes from your mother and one set from your father. And that's drawn in this. I had this great slide that I used in Europe, but I ended up deciding not to do it. It was an Etsy slide, and someone had literally crocheted eggs and sperm, and I was like, people have too much time, right? So you get your mom's DNA through the egg and your dad's DNA through the sperm, and that means you have 2 copies of every single chromosome. And the thing that's interesting is your body knows which strand of DNA came from your mother and which strand of DNA came from your father. This is something only mammals do. So other animals do not do this. It's just us, the primates, any milk-giving animal. And we have 23 pairs of these chromosomes. So 23 from mom, 23 from dad, 23 pairs. Fun fact, this is actually my son's chromosome. He's here with me, he's about 6'2". And I've been using these since he was born because I got this at one point and I'm like, good, chromosomes, I can use them. The one we really care about is this one because that's chromosome 15. And that's the one that's affecting our loved ones with Angelman syndrome. And we can draw the chromosome looking like this. You've probably seen these drawings before. What this says to like a geneticist is here's one arm of the chromosome, here's the other. This is what's called the center point. Clearly it's not in the center, but it's important for how cells divide and things we're not going to worry about. What's really important is this region here in red, because this region contains the gene called UBE3A. And genes are essentially instructions for making proteins. And proteins you can think of as machines. So your cells have all kinds of proteins and they all have jobs to do. Some of those proteins hold your cell up, some of them make them in certain shapes. Some of them keep your skin from being permeable so that you don't dehydrate to death. You will dehydrate to death if your skin is impermeable, if that gets messed with. And the way they do this is the genes on the chromosome, that's the DNA, They have a code. They have an alphabet that's made up of 4 letters, A, C, G, and T. And it's the combination of those letters in the order that they're put in that provides information to the cell. And the way the cell uses that information is it takes this DNA sequence and it copies it into another strand of ribonucleic acid, RNA. So DNA is the chromosome, RNA is the message that's made from this language. It uses a similar language, except it doesn't have T's, it has something called U's. This RNA is recognized by proteins that are machines, whose specific job it is to find these RNAs and make proteins out of the information they have. Okay, so this is what's called the central dogma of molecular biology. DNA is used to make RNA message is used to make proteins. Proteins are needed to do all of these steps, so there's this whole big cycle. But I want to stop here and just make sure, does that make sense to everybody? So your DNA has the message in it. To use the message, you have to make it into an RNA, and then the RNA has to be translated into a protein. And that's why we use those words. So we use transcribe to make DNA into RNA, right? You're transcribing the message, and then you're translating the message into protein. And that's why we use those 2 words. Okay. So typically, this is what happens, but only in the brain. So this is something I think we don't talk about enough. UBE3A is expressed throughout your body in every single cell in your body. All the cells of your body don't care if it's moms or dads. They express it off both copies. They use it however they use it in other cells. But our neurons in the brain do something really strange, and it's only those cells. So the cells in the brain know which one is mom's and know which one is dad's, and they turn UBE3A off on dad's chromosome. These are the only cells where this happens. We're not completely sure why. Like, sometimes we ask why. Like, why on earth would you do this? I don't know what the reason is. But UBE3A on dad's copy is turned off completely. It's like the message isn't there, the information's not there, nothing's there. It is there, but the cell doesn't see it. It only uses mom's copy to make UBE3A. And this is where it becomes problematic for us because of Angelman syndrome. So I do want to stop for a second and talk about what UBE3A is. As I mentioned, these are proteins, and proteins, you can think of them as machines. UBE3A's job, literally, is to find proteins that shouldn't exist anymore and get rid of them. It's a recycling mechanism. So UBE3A is this protein, and what it does is it has information that it uses along with these other proteins called E1s and E2s to find proteins that should no longer exist. They're too old, they're not functioning right, whatever the reason, and target them for recycling. So it's a very, I love that this protein is green in this diagram. It is a green protein, it is a recycling protein. Excuse me. What it does is it transfers a little chemical called ubiquitin. It's called ubiquitin because you can find it everywhere. Scientists think they're funny, so that's sometimes why you get these names. Ubiquitin is this little chemical, and UBE3A's job is to put that chemical onto proteins because that is the signal that that protein needs to go away. This big other structure of proteins is called the proteasome. The whole job of the proteasome is to take those marked proteins and chew them up so that you can use those amino acids to make other proteins. It's truly a recycling mechanism. And so you can imagine if you don't have this gene, there are proteins that are sitting in your neurons that shouldn't be there. There's too many of them, they're built up, they're not functioning well, they're garbage. And they're flooding the system and they're making it hard for the neuron to operate properly. Okay, I'm going to stop here and just say, are there questions about this? Does this make sense? Very few nods, so I'm thinking it doesn't make sense. I think the thing to take away here is if you don't have UBE3A, your neurons can't function the way they're supposed to. We used to say this, it's not the best analogy, but it's not a bad one. If you think about having Angelman syndrome, it's like having a car that's not tuned up properly. All the pieces and parts are there. They're just not functioning correctly, they're not coordinated, they're having some issues getting the signal through. So then how do we get Angelman syndrome? So this is the genetics. The most common way we get it is that you get a deletion that just removes that region on mom's chromosome that has UBE3A in it. This is what my daughter has, is she has a chromosomal deletion. I will say that we've always said that this is the most common form of Angelman syndrome. We're starting to realize as we do more and more genomic sequencing that it's probably going to be it's not going to be less common, it's just that UBE3A mutations are starting to become more and more common. And so it may turn out that deletions are not the prominent way, it's just the one that we detect the easiest. Okay, so deletion. How many of you know that your child has a deletion? Pretty decent number, okay. Hopefully I'll come back to this, but if I don't, I want to say it now. When I heard this, and again, I'm a scientist, I got so upset because I thought all those chemicals I worked with, I did this, this was me. And the doctor saw my face, I think, and she said, you didn't do anything. She's like, you didn't do anything. And this might or might not be comforting. If it is, it comforted me, so hopefully it'll comfort you. These deletions that occur in the chromosomes most often occur when you're making the whole set of eggs that you're going to have for your lifetime. That happens when you are still a baby in your mother's womb. So the deletions that led to your kids probably happened before you were even born. You didn't drink anything, you didn't walk next to any, you did nothing, right? You weren't even a gleam in your mom's eye when that happened. So you just have to keep in mind that DNA is really agnostic. It doesn't think it just does what it does. And these deletions occur because there's a defect in how the DNA replicates. It doesn't have anything to do with anything that you did. Hopefully that's comforting. Whoa. Oh, this is to show you the deletion. We think about this deletion as being huge, right? It's considered a micro deletion. It's actually small. You can't see it. Even if you looked at the chromosomes like I showed you before, it's tiny. It doesn't feel tiny to us, but it is. It's about 6 megabases, which sounds huge, but you have to keep in mind that our genome is 3,235 megabases big, which means if my entire outfit was our genome, we're missing a little tiny piece of the hem that you can't even see. There are a lot of genes in this region. So this is the class 1 breakpoints. So if you have a class 1 deletion, some of you may know the class, you may not. Class 1s are slightly larger. They take out a little bit more. Class 2s are slightly shorter. But the point here is that both of these common classes take out UBE3A on the maternal chromosome. They also take out these genes called GABA-R components. This is the GABA receptor. The thought is not having enough GABA receptor is probably contributing to our kids having epilepsy. Kids that have mutations just in those subunits have a really strong epilepsy phenotype and other developmental disorder. So that probably is not helping our kids, but it's also probably why our kids respond so well to benzodiazepines. So if you're taking clobazam or Onfi or Valium or any of those things, it's probably because we don't have enough GABA and those drugs up that response. Okay. The next mechanism is mutation. So this is when there is a change in the information that makes the protein not work correctly. So this is just another way to think of this. So I was telling you that you have the DNA that gets transcribed into an RNA, and then this RNA has information in it. And how the cell reads this is it reads it in 3-letter codes. So these 3 letters make words for a cell. And so like if it reads ACC, it says, okay, I need a thymine in that protein. And if it reads GUG, it says that's a valine. And CUU, that's a leucine. So when your proteins are reading this, they're like this amino acid, this one. They link them together. And you can think of it like information in a book, right? Let's say this is a sentence in a book. And normally this sentence says, the UBE3A gene encodes a protein called a ubiquitin ligase. Right? A polymorphism is when there's a change in the gene, but it doesn't actually affect the protein. And this happens because there are multiple ways to make T There are multiple ways to make L, there are multiple ways to make G, so sometimes you just get these changes, but they don't really do anything. But if you sequence, so if I sequenced UBE3A in every one of us in this room who doesn't have Angelman syndrome, they would all look different because the information can change without really affecting the protein. If I sequenced the gene of all of us in here for hair color, they would all be very, very different because we all have different hair color. It's still hair, it's still color, it's just different. Okay, so in this case, this would be a change where we've changed this piece of the DNA and RNA, so it's a different message, but it actually still encodes the same amino acid, so it doesn't change. This is really the same sentence. Some mutations will impact the meaning of the protein. They will impact the information. So in this case, we've made this change, and we've completely changed this amino acid. So now it's not leucine or L, it's turned into P, which is proline. This is different. Now this protein is not going to fold correctly, it's not going to have its machine in the right place, it's not going to function the way it's supposed to. And so if you think of it in our sentence analogy, this would be like saying the UBE3 gene explodes a protein called a ubiquitin ligase, which makes no sense. So we call these sometimes missense mutations because it's changed the meaning of the code and the protein now can't function. And then the last one I want you to know about is something called a stop mutation. What happens here is that the amino acids, the RNA sequence gets changed, and it gets changed in such a way that this information tells the proteins to stop making this protein. So the RNA is being read, you're making the protein, you're adding your amino acids, and then you get the signal that says you're done. And you say, okay, I'm done, and everything falls away from the mRNA and you don't make any more protein. The problem, if this happens, is now we've made part of the protein, but not all of it. And that means the protein's probably not going to function correctly. So stopping here, any questions about mutations? What kinds of mutations occur? How they affect things? Okay. Yeah. So she's asking if you got a genetics report, should it be able to tell you which one of these they had? Yes, depending on what they did. So if they did whole genome sequencing or whole exome sequencing, yes, they should be able to tell you exactly. Or if some of you might have gotten diagnosed much earlier, like I did, in those cases, if they did the sequencing, sometimes they will just, they'll tell you it's fine, but they won't tell you the information, but you can get it. But if you have your genetics report, there are plenty of geneticists around that could look at it and tell you. All right. Oh, well, I'm sorry, I should have said that. One more type, because some of you might have this one too. Sometimes if you add in extra pieces of information, like if you add or delete some of this information in the RNA, it changes the code because it's 3 letters. So if you remove a nucleotide, it shifts the code. If you add nucleotides to it that shouldn't be there, it shifts the code. You make a protein that has nothing to do with the protein it should have made originally. These are sometimes called frame shift mutations, small deletions. Sometimes this gets confusing as you might be told you have a deletion only in UBE3A. Your child is not deletion positive, your child is mutation because it only affects UBE3A. Happy to talk more about that if anybody feels like that's what they're dealing with. Okay, the other 2 have to do with cells knowing if your gene is mom's or dad's. For UPD, this is actually kind of miraculous. What happens in UPD is that when the chromosomes are being separated as things divide, somehow mom's chromosome didn't make it into the egg. This is not good. You can't survive without your chromosomes. So what happens when that egg gets fertilized is that dad's chromosome is recognized and the cell says, I need 2 of these, and it just makes a copy of it. So you have 2 chromosome 15 so you can be alive, which is really important. The problem, though, is that they're both recognized as dads, and so neither one of them expresses UBE3A, but it's there. The last kind that we talk about is something called an imprinting defect. In this case, the information that is used to determine if it's mom's chromosome or dad's chromosome is not correct, and so the cell can't tell apart mom and dad's chromosome. So basically the cell assumes that this one is dad's, even though it is mom's. So there's nothing genetically incorrect except for the fact that the cell can't tell that one's mom and one's dad, and so it just shuts them both off. Okay, questions about mechanisms? Yeah.
Are for just, I'm sorry, the mutations that you described are just for the mutation type of Angelman's?
Yes, so the mutations
Okay.
are just in
So
UBE3A.
the deletion is just, like, it's just not.
Deletion, the information's not there.
Okay.
The page just got ripped out of the book. Yeah. Good questions. Other questions? You have a question? Anytime, hon. Any more? Okay. All right. How do your cells know which one is mom's and which one is dad's? That seems weird, right? The way they do this, so like I said, in any cell that's not a neuron in your brain, they don't care. And so in these cases, they express UBE3A off both chromosomes, and UBE3A has jobs in all your other cells. Now you might be saying, well, wait, my kid has Angelman syndrome, so what's going on there? Is that a problem? It's not. Because even if there's a deletion that removes this gene is on and it's working fine and it's taking care of all the functions that need to be taken care of everywhere else. The brain is where we have the problem, because in the brain, there's this other gene that gets expressed that doesn't normally get expressed. Only in the brain. It's called lots of different things. It's been called SNRP-N. It's called the antisense transcript. You'll hear it called ATS. This gene turns on and it starts to make its transcript from dad's chromosome, but not from mom's. And this essentially is like a train. It's really strong, and because it's moving really fast and it's reading the DNA to make the message, this gene has no chance. It can't actually start, because it's transcribed in the other direction. So DNA has two strands. Each one of them can encode different things. This strand could be important for UBE3A. This strand is important for that antisense. The antisense is stronger and it wins. It basically runs through the whole DNA area. You can't make that much UBE3A because this train is running through the locus, proteins off. You make a little bit. If you look, all of our kids have a little bit of UBE3A from that paternal chromosome. It just can't win. It just can't get enough made because of this antisense transcript. Mom's makes it just fine because this antisense transcript isn't being turned on. And so if you're deletion, you've got this nice big strong train going through up here, so no UBE3A, and you don't have it down here at all, so no UBE3A. But this becomes important because this is how we're thinking we're going to do therapeutics, is we're going to basically stop that train. And we can talk more about that in a bit.
Hey guys, it's Amanda here. As you know, we're here for you, our community. If you have questions about any topics we can discuss in our podcast, or if you have ideas for future episodes, we'd love to hear from you. All you need to do is email me at dearamanda@angelman.org. Thank you so much for listening and your continued support of the Angelman Syndrome Foundation.
The way your cells know which one's mom and which one's dad have to do with a chemical called methylation. There's a different chemical on dad's chromosomes that was put there in the pathway to make sperm, and there's a different methylation pattern on mom's chromosomes when the egg was being made. And so when those two chromosomes get together in the fertilized egg, they have different chemical structures, and those structures get maintained every time that cell divides. So that's how the cells know which one's mom and which one's dad. Questions about the train, antisense? Anything good? Okay. All right, so this was the first time that anyone realized that genetic imprinting had anything to do with human disease. And so what I was just telling you about different chemicals on mom and dad, this is imprinting. People knew this was happening in mammalian cells. They knew that for some reason mom and dad's chromosomes were being treated differently. But this was the first time they realized it could cause disease. And this was Mark Lalonde's paper, where he found that people who had 2 copies of mom's chromosome 15 had something called Prader-Willi that was really well recognized. But other people had 2 copies, but it was dad's chromosome 15, and they had a completely different syndrome, and it turns out it's Angelman syndrome. And for a long time, it was like, why would that be? And it turns out it's this imprinting thing, because having 2 moms or 2 dads' chromosomes in your brain causes a problem. Okay. The numbers are up here, but like I said, these are going to change. This is the easiest to detect, so this is where we've done most of the diagnostics. We're getting more and more of these because we're just going straight to whole genome sequencing to see what's happening in children. This one can be detected pretty easily with something called a methylation test. You look to see if you have mom's and dad's methylation pattern. And that's how you find this one too. Because in this one, the methylation pattern isn't established correctly, and that's why it doesn't know. Okay. All right, so what does that mean for Angelman syndrome? So I want to show you this list of symptoms. These are the ones that are considered consistent. If you have Angelman syndrome, consistently you will predominantly be nonverbal. You'll find plenty of kids here that have words and talk, but mostly it's nonverbal. There are movement and balance issues. That's pretty consistent. There's some delay in your skill development, and there's an abnormality on your EEG, and that's the electrical recordings from your brain. You may hear about this called delta power. So we know that there's a specific electrical signal from the brain of a child with Angelman syndrome that looks different than anyone else. And in fact, that's how my daughter got diagnosed. So she was seizing. We were in the hospital. I've got a picture of her at 11 months old with the whole head wrap thing and the look on her face like, why am I here? And she didn't, of course, sleep all night. She was up all night. But an intern in the back never saw my child. He saw the EEG that was being transmitted to the room, and he said, oh, I know that one. That's Angelman syndrome. And the geneticist came back in the room and said, I think we're going to test your kid for Angelman syndrome. And it was all because of that EEG. She also told me that my kid didn't look like a kid with Angelman syndrome. And you've seen pictures of my kid. She looks exactly like a kid with Angelman syndrome. I don't even know what that means. Okay. Frequently, our kids have seizures. And again, it might be more common in deletion because we have that involvement of those GABA genes. Associated. Not all kids have these issues. Some do. There's the anxiety, sleep issues, GI issues, feeding issues, drooling, mouthing behaviors, aggression, myoclonus. This is not consistent, right? Some of these you'll have, some of these you won't. Just depends. So, and in general, this is very general. Don't definitely take this with a grain of salt. These slightly larger deletions, and actually there are some that are even bigger that are more severe, these tend to impact our loved ones the most, and mosaic forms impact them the least. So you might say, what's a mosaic? A mosaic is when, if you look at all the neurons in the brain, some of them are missing UBE3A and some of them aren't. And we know from those individuals, because a lot of them do speak in sentences and can do things with a lot less difficulty than our kids can, that we don't need that much UBE3 in the brain to get some really nice results. So if you're mosaic for any of these things, it might be less impactful. And I say this in general because I have met plenty of kids with mutations in UPDs that have horribly uncontrolled seizures. I have met some class 1 kids that you would think have to be mutation because they're just functioning fantastically. It's all grain of salt. And you might be saying, well, then what's causing all the symptoms that my kid has? Why is it so different? Why are all these kids so different? There's 2 things. So we know all of the unique symptoms of AS are because you don't have UBE3A, all of them. But keep in mind, there's 20,000 other genes in your body that make proteins, and all of those are important for who you are. And so we're missing one, but there's 20,000 others that are in there. And so, for example, some of you might have kids that are way paler than you are, right? My daughter is white, blonde, blue-eyed. And that's because there's another gene in that deletion that encodes for pigment. And if you lose both copies of that gene, you have something called ocular albinism. I have a friend whose daughter had perfect pitch. Her parents were musicians. If anybody had an instrument that was out of tune, she would put her hands over her ears and leave the room, right? That's not UBE3A, that's all those other musical genes that she got from her mom and dad. So you know this, right? Your kids have colors they like, they have sounds they like, they have bands they like. That's not UBE3A, that's the other 20,000 genes doing their job. Okay. We actually do have therapeutics for Angelman syndrome. Everybody's really big on this, like it's a big buzzword for money, right? We need therapeutics, we need therapy. We have them. They're not the ones we want, but we have them. We have really good anti-epileptic therapies. In fact, so good that Ron Tebert was asked to do a trial for the new drug Epidiolex. None of his AS patients were bad enough to qualify for the trial. So none of them got into the trial because their seizures were under control, which is good. We have some things that work for sleep. They don't work perfectly, but they work. Some of them is the melatonin. There's some other drugs that work for sleep. There have been some behavioral therapy studies that were funded by ASF that work, but they're hard. I'm happy to talk to anybody about them if you'd like to know about them. My daughter went through this when they did that trial. She was 5 at the time. By the time the trial was over with, she slept through the night and has continued to do so since she was 5 years old, but it sucked. So, but happy to talk to you about it. We have reflux medications. We have some behavioral therapies. We have some drugs that can help with anxiety. We have physical occupational therapy for the motor issues, and we do have communication therapies. We have the iPads, we have education, we have speech therapy. These are all things that are important and they work. One thing that is maybe the silver lining for us with Angelman syndrome is that our kids can learn. It might take them longer, they may need more repetition, but they will learn. And so any of these things we can do for our kids, they're beneficial. And our kids are smart. And you know this from your own kids, right? The things they get away with, the clever little tricks they pull, that is smart. Okay, so our priorities for your child, you know this, health. Seizures, sleep, reflux, GI, anything that's keeping your kid from being 100%, you want to take care of that. We know from studies that have been done, reflux and GI issues, especially constipation, will cause your seizure threshold to go down. So if you're having trouble with seizures, you also want to take care of these other things because they will make it harder to get your seizures under control. Communication. All ways of communicating, everything works. If your kid makes a sign, encourage it. If your kid unfortunately, my kid makes a pig snort when she wants something, it's annoying. It's very effective because we will come running to make her stop making that noise. And so she trained us and now we're stuck with it. But we're not gonna tell her not to make the noise, right? Because when she does that, it means my movie's over, I'm hungry, this toy sucks, I want something else, it's really helpful. Behaviors including sleep, anything you can do to get those behaviors under control. Someone told me early on not to do anything with Sophie I didn't want to do for the rest of my life. I forgot about that when the pig snort started, but they're right. So anything, especially when your kids are little, you can do so much more than when your kids are adults and are very autonomous and don't want your input, which is appropriate for an adult. You can find record keepings that work for you. I think this is one of those things that makes me crazy even now. I have too many doctors, I have too many medicines, I have too many, when they're like, what medicines is she on? I should be able to tell them, and I'm like, I don't even remember anymore. I'm like, she's on this, I'm having to go through my head of opening each bottle and which pill it is. Just make your life as easy as you possibly can. Because for you, your health is the priority. Everyone says this, you can't have happy children if you don't have happy parents. And what they mean by that is if you're not taking care of yourself, you certainly can't take care of anybody else. And you know that whether it's taking care of your individual with Angelman syndrome, or maybe taking care of a parent who's ailing, or a friend who has cancer, any of those things, if you're not healthy, you can't do it. So this is really important. And don't feel the least little bit guilty about doing it. You gotta get some sleep. However you do it, you gotta do it. I don't know how I didn't kill someone in a car the first 5 years of Sophie's life because I had no sleep and I drove around, and apparently that's worse than being drunk and driving, and I feel really bad about that now. And you have to have good community, and that means supportive community, right? Not fighting, cliquey, crazy community. It has to be good, supportive community. Be thoughtful about social media. And the reason I say this is I had to take a break from Facebook because it was killing me. It was killing my mental health. It was making me miserable. And this is how it works. And I know this is funny, but it is how it works, right? Someone will say, I don't like mangoes, I like oranges. And then you get these responses. Oh, so you hate oranges. You didn't mention pineapples. Oh my God, get over yourself. I'm so angry with you. You know, it's like, I just like mangoes. It's nothing. And it gets really bad around politics time because we all have very strong feelings about that. And that's totally normal. It's not normal to fight with people in cyberspace about it. It gives the illusion of connection without the actual benefit of having a connection. There's plenty of research that shows actually physically being near somebody is healthy. Connections on the internet, sometimes it's the best we can do because our lives are the way they are, but it's not as beneficial to you. And this is the thing that really killed me, and this is why I took my break. Studies show that even if every single thing you see on Twitter or Instagram or Facebook is positive, it still will cause depression in you. We don't know why. Okay. Resources that are on the websites, lots of things that you can do. People don't look at websites anymore. I don't look at websites anymore. If you need a resource and you can't find it, you can send an email to any of the staff at ASF and they will point you to the page. They're gonna hate me for saying that, but they'll do it, and you need to find the resources. Okay, the last thing I wanna leave you with, unless you wanna have some questions, is words really matter. And I say this because we are the family, and we can educate the clinicians on how to better talk to us and talk to our kids. This is what I would prefer people to say. My daughter, Sophie, is strongly affected by Angelman syndrome. I know other kids who are less affected by Angelman syndrome, right? That doesn't feel bad. That just feels like a statement. This feels bad, right? If someone tells you your kid is severe or low functioning, that feels really bad. But this is a lot of times the message that clinicians use. These are clinical terms for them. And they don't realize what it's doing to you and the way you're feeling about your child. So I was at a scientific meeting for my work, which has nothing to do with Angelman syndrome. And there was a session that was going to be about rare diseases, and they were going to present something from the Angelman syndrome natural history study. And I thought, I'll go listen, right? And I get in there, and the person who was presenting was giving a case report. And she starts the case report, and she's like, whoop. Seizure started around 7 months, sitting without support when she was 3 to 4 years old, can maybe sit for 30 minutes without protection, only can walk if you're holding her up, has tried 6 seizure meds, is currently on 2, had the lowest possible score on the Baileys for her age, and her conclusion was this child is so cognitively impaired that she is the most severe functioning Angelman syndrome child that they had seen. She said other things that made me realize she's talking about my kid, right? So I'm in the room and she's talking about my kid being so intellectually impaired. And I was mad, and I realized at that point that like I'm not thinking straight because, you know, I know my kid, and I'm like, that's not my kid. I mean, yes, that's my kid, but that's not my kid. So I want to show you this incredibly low functioning severe child. Hopefully can you play the movie? Because I probably can't. Okay, here she is when she's about 8, and this is her bike, and we used to go to the park and just let her go because she likes to ride around on her bike. And you'll hear me talking to her because if you notice right here, there is a young guy walking in with his little toddler, and watch her, she's casing the toddler and she's slowing down because she likes to hit people with things, especially toddlers because they make great noises. So now if you watch her, she's casing that kid. Every time she goes around, she's looking where the toddler is. And I'm watching her because I know what she's doing. So she's like, haha, there's the toddler. And now she goes after him, and I have to stop the video and go get her. She's not low functioning. She is very strongly affected by Angelman syndrome, no doubt. But that is not a kid with an intellectual disability that makes her the strongest or so debilitated that she's essentially a houseplant. And that's the way they were talking about her, and it was really painful. So I tell them all the time, please don't do that, right? Especially when you've just gotten diagnosed. And the first thing, when we got diagnosed, there was some kind of mix-up and they didn't tell us. And we went back to see the neurologist as a follow-up from that stay in the hospital. And he said, well, now you know it's Angelman syndrome. And I said, what? And he went, didn't the geneticist call you? And I was like, no. And he turned white. And I think because neurologists don't do that, right? They don't tell you genetic diagnoses. So he stopped and he said, okay, she has Angelman syndrome. And I might have been the only person in the world who was happy about this, because at that point, all the symptoms my kid had, I had a list of things that were possible that she had. This list were all things that were gonna kill her, and this list were the things that weren't. Angelman was on this list, and I was like, thank God, she's not gonna die. So he gave us the result, and he looked at me and he said, you know, nobody's written the book on Sophie. Only Sophie writes the book on Sophie. You have no clue what this is gonna be, but here's some resources, and he handed them to me. But I remember thinking at the time, thank you for saying that. Because if he had said to me, this kid is gonna be so low functioning that your life is gonna be miserable, I don't think I would have walked out of there in a good mood. Okay, questions about genetics really quick, and then I'll tell you in 2 seconds why we think we can have good therapeutics. Questions? Yeah.
So you mentioned that we need very little protein of UB3A to function like angels need. Do the paternal chromosome, which is silenced, leak any of that?
Yes.
Because that's what I've heard, and leak is for the better use of word.
Yes, it does. So there is a little bit of a leak off that paternal chromosome. It's just not enough. And one of the strategies that is still out there that we can still think about is how do we stabilize that little bit that gets made so it sticks around longer and does its job. That could help. But yeah, there's a little tiny bit that gets made. We just need more of it. All right. Here are the major strategies, and hopefully this will make sense. The two major strategies for fixing, and I say fix and I don't mean fix, I should use a better word. What I mean is ameliorating, which is a big fancy word, but basically how can we make AS better for our loved ones? How can we make it affect them less? That's really what we're trying to do. We can just replace UBE3A. So our kids with Angelman syndrome are missing UBE3A. Either it's not there or the gene just doesn't function correctly, the protein's not working correctly. How do we fix that? Well, we can take a virus, this is called adenovirus or AAV, and we can just put UBE3A back in. So we can infect the brain with the virus. We know how this virus works. It'll go to all the cells it can reach. It'll put a message in there to make UBE3A, and it'll start making the protein. Yes? This approach would work for any of the genotypes, any of them. UPD, imprinting, mutation, deletion. Because what we're doing is we're basically bypassing the chromosome and we're saying we're gonna put it back in and we're just gonna express it somewhere else with different controls. And that way it'll just be made. And our hope is that it'll be enough and it'll be in enough places that it will provide some relief. The problem here is that the virus doesn't actually hit every single place in the brain. So if you inject, it'll hit a lot of them, but not all of them. And so you have to worry about how well can you get it distributed, because we're not really sure which regions of the brain are the most affected by Angelman syndrome. If we knew it was just this little piece over here on the right, no problem, we'll just go hit that one. But we don't know that. The other way you can do it is the way that are right now in the clinical trials, which are these things called antisense oligos. This is a method where basically what we're doing is we're gonna trick the paternal chromosome into turning that little leaky bit of expression into strong expression. So now we can get UBE3A strongly in the neurons where we need it. The other strategy is to try to correct what's going wrong with the neurons without touching UBE3A. So like I said, without UBE3A, there's probably a lot of garbage that's building up in the neurons. They're not functioning correctly. Proteins aren't being turned over the way they should be. If we could figure out what is actually going wrong, we can maybe fix that instead. Maybe that would be easier to fix. Maybe there's already a drug out there that does that if we knew it. The reason we think this is gonna work, besides the fact that we're seeing good results, is there was a paper published in 2007 that got all of us super excited. And it basically said, if you make mutations in this other gene that we know is really important for how neurons function, and you make that mutation In a mouse that has Angelman syndrome, that mouse doesn't look like it has Angelman syndrome anymore. So if you mutate something else, it can compensate for not having UBE3A, which is crazy, but it worked. And the thing that was really interesting about this is that this gene that they mutated is called CAMK2. CAMK2 doesn't even turn on in your brain until after you're born. So it says that you can develop with Angelman syndrome and then turn this gene off and not have Angelman syndrome. So it's not developmental. It's not like your brain didn't develop while you were in utero. It developed probably fine. It's just not functioning correctly. And we found that in early studies, if you just put UBE3A back with that virus, if you just put it back in the mouse brains, you can rescue quite a bit of the phenotypes that hopefully you heard Dr. Ipa Elgersma talking about. Mice can't run on a rod very well. I don't know what that's really analogous to. They can't bury marbles. Again, I don't know what it's, but they can't do these things, but you can do it if you put that virus in and put the thing back. IPA has shown that some things can only be rescued if you restore UBE3A early, but a whole lot of things can be restored no matter what the age of the mouse is. And that is really good news for all of us whose kids are not 2, because maybe we can actually get some relief of the symptoms and the effects, even if our kids are 30, 40, or 50. Okay, this is the bottom line though with all of this kind of work. If you are a mouse who has Angelman syndrome, we have very good news for you, because we can fix it. That doesn't mean that we know exactly what we need to do for humans, because mice aren't humans, right? What they do is much more simple. They don't have the complex things that we do. They don't have the complex communication we have, although we did hear at the research session, they have a pretty complex ultrasonic squeaky thing they do when they talk to each other that is affected in Angelman syndrome, so maybe it's analogous. But we believe that these therapeutics are possible, and we're not going to know until we know, and we need to do clinical trials, which is what we're doing. I'd be happy to talk to you more about clinical trials, but I think we're sort of at the end of time. So I'm happy to take any questions that you have. In terms of the trials that are going on, again, the industries are going to do some updates today about where they are and what kinds of things are happening with their programs. But after that, I'm open for anything that you have or anything that hasn't been clear. Yes?
They love the technical details, but even other friends who are very educated, I guess I would say, I want to know if it's wrong to say, okay, well, Angelman's affects the proteins. Basically, proteins aren't being kicked out. And so, like you were saying, there's a lot of garbage. But if there were just like a couple other just basic layman's things to say to somebody who doesn't understand this at all. We have a lot of friends that know our daughter quite well. They're even scientists, and some of them are even, like in labs doing things. And if I had just a couple sentences.
I was trying to see if I had something here. What I tell Sophie's teachers, because they look at Sophie as a whole laundry list of things she can't do. And what I always say to her teachers is, look, her brain is fine, but the neurons in her brain can't function optimally. They can't talk to each other optimally. Essentially, it's like some days there's a big snowy TV right in front of her face because nothing's working. She can't get it into focus. She can't hear you. She can't see. She's just going to sit there today and play with her fidgets because she can't do it. Other days, there's some play in that screen and there's some focus and things look better, and those are the days where she's learning and she's on and she's ready. In Angelman syndrome, we're missing a gene that lets those neurons function optimally. And so our kids are basically functioning where they have to try twice as hard and work twice as long to get the same effect. I don't know if that helps you. And if you're thinking about like, what's the actual underlying mechanism? They're missing a gene that's important for regulating all the proteins in the neuron that lets the neuron function the way that it should. So, yeah. Yeah, she's saying it has something to do with GABA. We do know that we have some hints that not having UBE3A means that there's a protein that basically sucks GABA. Have enough GABA. And you also don't have enough GABA receptor if you're a deletion, so you're missing this important signal that the neurons use to talk to each other. So that doesn't help. But again, that's why things like Onfi and clobazam seem to work so well for our kids, because that's exactly what it does, is it puts more GABA, it keeps the GABA around longer, and it lets the receptors hear it clearer. So it's not perfect, but that's why it will sometimes work. Some kids it won't work at all. So we don't know that either. So does that help? Okay. Other questions? These are good. Yeah. Here, he's going to give you the mic.
I'm very interested in clinical trials and was wondering if you have any contact information or somewhere that we can contact and reach, because as you said, there are you all are working on things, and especially the virus, that is quite fascinating actually. So would like to know more about it.
So
Thank you.
with the virus, we're not there yet. So we're going to have to do a lot of work to make sure that we have a virus that's nice and safe and does what it needs to do, that the FDA is going to let us, because since our kids have a normal life expectancy, we're not going to do anything that's not safe, right? Ethically, we can't do that. We're not there yet. But with the ASO trials, those are happening now. And there are ways to contact. So we have a website that is Angelman Clinical Trials that we can make sure everybody gets the thing to. It's run in conjunction with FAST and ASF Canada and ASA, so that we have all the information there about the clinical trials and what's happening. There's also a website called clinicaltrials.gov, G-O-V. Every clinical trial that's happening in the world is on clinicaltrials.gov. So you can go there and look, or in the US, I should say. You can go there and put in Angelman, and it'll pop up all the studies that have been done with Angelman or that will be done with Angelman. The other thing you can do is go to the clinical trials update today, because those representatives will be in the room, and you can ask them, are you going to recruit more people? Where can I sign up? How do I get in line? And one thing I think you can be proud of as a community is that we're a rare disorder, and therapeutic companies are always like, this is going to be a problem because we're not going to have enough people to be in the trials. None of these trials have had any problems recruiting enough people to be in the trials. And I think that's because we have a really active, thoughtful, scientific-minded community, and people are willing to participate. And for every one of you that is participating or can participate, thank you. Because since my daughter does not walk independently, she's excluded from all of the trials. So she can't participate. So I'm relying on all of you to get through the trial so I can get her some medication at some point. If you have more questions, I'm around all week. Just grab me. I'm happy to chat. Thank you guys so much.
Thanks for listening to the ASF podcast. If you enjoyed this episode and would like to support us, please share it with others, post about it on social media, or leave a rating or review. And please don't forget to subscribe.